Hello, everyone, and welcome to today's live broadcast, Clinical Laboratory Workflow, the Impact of the 3 R's, presented by Dr. Jeannine Holden. We're excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Beckman Coulter. Beckman Coulter is dedicated to advancing and optimizing the laboratory. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life science researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. For more information, please visit www.beckman.com. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice, you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce our speaker, Dr. Jeannine Holden. Prior to joining the Beckman Coulter team in 2013, Dr. Holden was Associate Professor and Director of Hematopathology and Flow Cytometry at Emory University School of Medicine in Atlanta, Georgia, where she ran a hematopathology reference laboratory providing expert consultation service in microscopy, flow cytometric immunophenotyping, fluorescence in situ hybridization, cytogenics, and molecular diagnostics. She currently serves as past president for the International Clinical Cytometry Society, ICCS, and is actively involved with both the International Society for the Advancement of Cytometry and the European Society for Clinical Cell Analysis. She is also past chair of the ICCS Advocacy Committee and serves as the ICCS liaison to the College of American Pathologists Diagnostic Immunology Resource Committee. Dr. Holden's combined deep experience in diagnostics, laboratory management, education, and regulatory and reimbursement strategy is unique. Dr. Holden received her undergraduate degree from the University of Virginia, her MD from Emory University School of Medicine, where she also completed training in anatomic and clinical pathology and hematopathology, and her MBA from Georgia Institute of Technology. Please join me in welcoming Dr. Jeannine Holden. I will now turn the presentation over to her.
Thank you, Christy, for that very detailed introduction, and thanks to all of you for attending today's webinar. It's my pleasure to speak to you today. The topic we're going to be addressing, of course, is the clinical laboratory and specifically the impact of what I call the 3 R's. So we're going to go ahead and get started. Just an overview of what we'll be discussing in the talk. I'll be coming back to these points as we address them. And this first slide, of course, is a little bit about my background, which Christy has already described in great detail, but just to say that essentially my career up until 2013 was in academic medicine, where I did both diagnostic hematopathology and operated a reference laboratory in that setting. And specifically, I have particularly deep experience in flow cytometric immunophenotyping, and that laboratory does about 6,000 leukemia and lymphoma cases per year. Since joining Beckman Coulter, I actually have 2 roles. One is Director of Scientific Affairs, and the other, I now head the application support for the developed markets, that is Europe, Canada, and the U.S. So we're going to start by describing the 3 R's and discussing in some detail their underlying drivers. So what are the 3 R's? Well, the first one is reimbursement. That is to say how much money laboratories performing testing will obtain, whether they're getting it from, say, insurance or a governmental agency. How is it that the laboratory funds its activities? The second is regulatory, and this is essentially the regulatory framework within which clinical laboratories have to operate. And the third R is resource. So when I say resource, I mean human resource, specifically those of us who actually work in the lab. So we're going to get right off with the audience participation portion of this session. So the question is, which of the 3 R's do you consider to be the most important? So just click one of these, either reimbursement, how much are you concerned about, perhaps about how much money you're getting, or think you will get in the future. Regulatory, are there regulatory issues that you think may be of concern to the lab? And resource. So pick one of those. We're going to leave that poll open for a few more seconds. And I'll take a sip of water, which is every time I ask you guys a question, I get to okay, so we closed it, and we're going to go ahead and see the results. Okay, very interesting. Excellent. So 21% of you feel that reimbursement is the most pressing issue. 38% feel that regulatory is the most pressing issue, and 40% feel that your human resource is the most pressing issue. So that is very interesting and excellent information for me as I go forward. Later, actually, we're going to see what another audience said when they saw this slide. So I'm going to continue from there. Okay. So with that, we actually are going to start, even though a minority of you actually thought that reimbursement was the most pressing issue. I am going to talk about it in some detail. And I'll point out that a lot of people don't understand how their laboratories actually get money, and that's why I bothered to describe it in the context of this slide, or of this talk, rather. So typically the 3 ways that a laboratory can get reimbursed for the work that they do. So this is essentially your revenue. This is your top line, the money coming into your lab. So one means is fee-for-service. There's also something called capitation, and finally what's called bundling. So in a fee-for-service system, each test or procedure, because this also can apply to things like surgeries or office visits and so forth, each test or procedure is reimbursed separately. So this is actually very straightforward when it comes to the laboratory. You know exactly how much you're going to get paid per test. You know that each and every test you do will get reimbursed. And you know that the more testing you do, the more you'll get paid. Now, so that's the pro to this system. The cons of the system are numerous. The first is that there is no gatekeeper incentive. There's no incentive for the laboratory to do less testing and to perhaps avoid doing things like repeat testing. And so for that reason, it's assumed third-party payers, therefore, will tend to assume overutilization even if you're not doing it. So plenty of labs actually do still say, oh, I got 2 samples within a 24-hour period. I probably don't need to do them both, or if I do, I should check. But the third-party payers will actually assume overutilization just because of the fact there is no built-in gatekeeper incentive. So there is this potential overutilization to essentially do too many testing. The problem with this, what happens typically when third-party payers assume utilization is overutilization, is they will actually put in place limits that, although the idea is to prevent overutilization, the ultimate consequence is that they actually may limit patient access. And this can be quite problematic in certain markets. Now, the second means that you can get, actually, before we go on to other means of doing it, I'm going to talk a little bit about flow cytometry because that's where I have particular expertise. And I'll point out that in the U.S., flow cytometry for assays other than lymphocyte subsets and stem cell enumeration, beta CD34, is based on nonspecific stacked, what are called CPT codes. And so for the first marker, let's say you're running, say a 20-marker panel. for leukemia and lymphoma, you would actually bill 88184 for the first marker, 88185 for each additional marker, and then depending on the number of markers you had run, you would bill one of the professional fees. So it's a fairly complicated system, and there are particular issues with it. There aren't actually a lot of pros to the system. There are a number of cons, however. Again, there's no gatekeeper incentive. So in this instance, the potential overutilization really speaks to too many markers. So what happens is that third-party payers, again, will assume overutilization and put in place policies that try to limit the number of markers that are actually being run, either by not funding or in some case arguing that you should be doing fewer markers when, of course, you actually should not. That's driven by medical necessity. The codes themselves are non-specific, so they are agnostic to the actual test, which is problematic if you're trying to figure out why the test was actually done. And it's also extremely difficult if you're trying to do cost accounting in your laboratory on a per marker basis, because of course we don't actually do the testing on a per marker basis. Now, another means of reimbursement is capitation. So under a capitation system, and this is typical of inpatients in the U.S. and many patients or all patients in healthcare systems in other parts of the world. Under this system, the tests are not reimbursed separately. They may be all linked to an individual patient, so you essentially get a set amount of money for each patient in the healthcare system, or they may be linked to a care event, and so that's the system we have in the U.S. for inpatients under the DRG system. The pros to a system like that is that they actually do encourage gatekeeping and lab efficiency, because the amount of money you have available to you is limited, and you will typically therefore want to not do excess testing. The cons, of course, is the lab bears the financial risk. If for some reason you end up having to do much more testing than the capitation model predicted, you may actually lose money. And it is also possible that the system may limit patient access as an unintended consequence. Now, the third system is bundling, and this is also something we are seeing in the U.S. now. In this case, tests are grouped, and these aren't the same tests. They just, the payer, often Medicare in the U.S., will simply assume that certain tests are similar enough that they can all be reimbursed at the same rate. Now, the payers love this because it simplifies their accounting. They don't have to come up with different reimbursement for every test on the books. They can say, well, this is a level 3 of this on this particular fee schedule, and all of the level 3 testing gets the same reimbursement. The problem, of course, is that there's the risk that the test will be assigned to the incorrect group. This actually happened in the U.S. a couple of years ago for outpatient flow cytometry testing, for instance, in which they were the test was actually assigned to the wrong group and was consequently way under reimbursed for a period of a year. The lab financial risk varies with the test mix. So if you do a lot of testing that is over reimbursed under this system, then your lab will do well. If you do more of a test that's under reimbursed within that particular group, and the payers acknowledge that this happens, then you'll be bear the financial brunt. And again, there's a potential for limiting patient access as unintended consequence. So actually, all three of the reimbursement methods run the risk of limiting patient access to testing as unintended consequence. So why are we doing this? Why are we limiting access? Why are we why is it that people that pay for testing are so concerned about reimbursement? And it turns out, of course, that there are big picture drivers behind this. We are seeing worldwide population growth. People are living longer, and as a consequence of living longer, not only are they alive long enough and have more time to consume health care, but as you extend your life, you actually require increased intensity of care. you have more time to develop cancer. Other diseases like type 2 diabetes, autoimmune disorders are increasingly frequent with age. And the types of testing that are being delivered to the patients is also more intense. You see molecular testing, for instance. High complexity ancillary testing becomes more common with these diseases, and of course the types of care. These patients are more likely to get biologics, for instance, if they're having autoimmune disorders, or say even cell therapy for some of these disorders, including cancer. So the goal for essentially society overall, and certainly third-party payers, is to deliver more care but keep costs from spiraling. So we don't anticipate that this is going to change, and in fact, here's an example of what's actually happened in the last couple of years for Medicare reimbursement for flow cytometry in the outpatient reference setting. So those CPT codes that I described before, so this is 88184, which is the first marker. You can see in 2004 how much the reimbursement was for in that particular care setting. It actually trended up a little bit, and then it is now in 2017 will fall dramatically. And we're actually seeing this across the board. And so these are actually some quite deep cuts. The overall cut for, say, a typical case, say somewhere between 20 and 30 markers for leukemia and lymphoma, will, if we stay on our present course, will drop 70% relative to the 2015 reimbursement. So we have another question. So we know that reimbursement is we expect reimbursement to fall. Which of these would be the most effective countermeasure for falling reimbursement? So you operate a lab. What's the part of your laboratory operating expenditure that you can modify in such a way that it will actually offset the decreased reimbursement? So your options are to decrease labor expenditure, you decrease your consumables expenditure. I don't give you an option to decrease both. You have to pick one. Or maybe you are a nihilist and feel that you can't actually affect either one, in which case you'd say not applicable. But I expect most of you figure that you can either decrease the amount of money you spend on labor or decrease the amount of money you spend on consumables, and thereby hopefully offset some of the decreased reimbursement. Okay. So that's great. Oh, interesting. 23% of you feel that you can't actually drive down either your labor or consumables expenditures. Now, it's possible that maybe you operate in a laboratory environment that's already maximally efficient, so we'll assume that that's the case. Either that or you're a bit of a pessimist. A third of you feel that the most effective means for falling reimbursement would be if you could spend less money on your consumables. And then 44% feel that you would have the biggest impact by decreasing your labor expenditure. So that's very interesting information, and thank you for that. Okay, so we're going to move on to regulatory. Now, for those of you in the audience who are not currently in the United States, you may not be aware of what's going on in the FDA. In October of 2014, the FDA issued a guidance. There's actually two guidances around this, but it has to do with what's called laboratory developed test. So most of the testing that is done in a clinical laboratory is, in terms of the types of tests, most of those are actually laboratory developed tests. They're actually not in vitro diagnostics. Now, in terms of the total test volume delivered, because some of the tests are so high frequency, something like a CBC or say a sodium potassium, serum electrolytes or something, of course those are all in vitro diagnostics. But if you look at the broad menu of tests that are actually delivered, most of those are actually developed. There is no in vitro diagnostic. And a good example of this, of course, for me is flow cytometry. With the exception of some kits for lymphocyte subsets and stem cell enumeration, almost all testing is done in the context of laboratory developed test. And this guidance essentially, the idea behind the guidance is to increase the regulatory burden essentially on laboratories that are doing this testing to make sure that they are actually doing the testing well, that they're designing them well, and that they've been correctly vetted and validated and so forth. And there has been considerable pushback against it. Generally speaking, any test you do would be assigned a risk category, and the higher the risk, so that's the higher the risk category, the higher the regulatory burden, such that something that might be a particularly high-risk test, your regulatory burden would approach the level that vendors who have to develop in vitro diagnostics currently have to have. And as you probably understand, the fact that there aren't more in vitro diagnostics for many assays has to do with the fact that it's very expensive to develop them, and a lot of that expense is related to the regulatory burden. The regulatory burden also lengthens the timeline. It takes much longer to develop a product if you're going to be marketing it as an in vitro diagnostic. So it's expensive in terms of the money, it's expensive in terms of the timeline, and it essentially keeps these products from coming to market. All right, so remember we had the question about which of the 3 R's do you consider to be most important? And this was actually, I gave a much shorter version of this talk at ICCS last month actually. And I asked this very same question. The question was, which of the 3 R's do you consider to be most important? Now, interestingly, the FDA was actually in the audience, so it's possible that drove the high rate of response to regulatory, but it does actually mirror this audience in that reimbursement was seen as the least significant, and regulatory and resource were both considered more important. So I'm going to ask another question. In your laboratory, what do you anticipate would be the impact on workload of increased regulatory oversight? So now this might be the FDA, or it might be, if you're in Europe, for instance, I would not be surprised to see increased regulatory oversight. We're seeing this change everywhere, especially if you're in the U.S. and you are doing testing like flow cytometry or molecular assays, anything that's a laboratory-developed test. Do you anticipate the impact on workload would be increased, decreased, or no change? So you have to take into account, more paperwork, more admin, and so forth. Oh, look at that. Well, not surprisingly, most of you felt that it would increase your workload. Some feel that it would decrease your workload, and some feel that there would be no change. But the overwhelming response is that most people feel that if they have to, that a higher regulatory burden is likely to result in increased workload. So I have a follow-up question. So which of these would be the most effective countermeasure for workload impacts secondary to increased regulatory oversight? So well, the last option is not applicable. Workload will not increase. So if you said you didn't think it would increase or that it would actually even decrease, you would, of course, choose that option. Your other options are you could perform fewer LDTs, so you wouldn't have the regulatory burden associated with them. The other would be to perform more IVDs. You could increase staffing, assuming you could find them, or all of the above. And of course, performing more IVDs is theoretical, assuming you could actually find an IVD that did the testing that you needed to do. We can assume that for purposes of this poll that you actually might be able to source those IVDs. Okay. All right, so in spite of the fact that many respondents said that the workload initially that they thought it would not that it would either not change or decrease. Most people feel that did not choose this option. So all of the above was the winner. People thought they might be able to take a multi-pronged approach, but performing fewer LDTs, of course, would have an impact. IVDs if available, and increased staffing. It gets the last option, although certainly if it were an option, you might have to do it. All right, so this brings us to resource. So human resource. So labor is the most valuable asset in any lab, certainly, but also in any enterprise. Anywhere you work, whether you work in a university, in industry, in retail, for instance, any service organization, labor is always your most valuable asset. And this is true even in say, factories where you've got a lot of automation. Your human resource is still typically the most valuable. It's also typically the most expensive as well. And to that end, when you think about the labor that you're asking staff to do, there's a difference between manual and intellectual effort. So manual labor, when it's done by people, is inherently error-prone. It's just the nature of the beast. That's what we do. We're essentially random event generators. We're also prone to repetitive motion injury, and this is a real issue for people that are in these jobs for long periods of time, especially. There's also this very important concept of opportunity cost. What could you be doing if you weren't pipetting? If you think about the many things that you could be doing, which include developing new assays, you're doing your QC, continuing education, there are lots and lots of things you can be doing when you're actually doing manual work instead. Now, what humans are really good actually is intellectual effort. And so typically jobs essentially are more highly paid, they're more valued as intellectual effort increases. Now, in the case of medical laboratory personnel, medtechs, ASCP has actually been studying this question for quite a long time. And they argue, and this is certainly true in my experience and from what I can tell the experience of others, that highly skilled workers in general, and certainly medtechs, are difficult to recruit and relatively difficult to retain. It's a tight market, and if you try to hire anybody recently, it can be relatively difficult. We also have an aging labor pool, and I will say I find this quote sort of funny. It says laboratory personnel labor force is aging at a 78% faster rate than the entire U.S. labor market. This does not mean that we're actually getting old more quickly than other people, which is reassuring. It just means that on average, we are aging faster. Basically means that young people are not entering the labor pool. And this is lab personnel overall, not just flow cytometry, but all labs. Now, if we look at laboratories overall, the vacancy rates do, for all staff, actually vary according to the type of lab. And flow cytometry labs tend to be relatively low, although most labs do report vacancies. But say compared to phlebotomy or a core lab, our vacancy rates are relatively low. And this is actually from 2014. 2014 was the first year that they actually included flow cytometry as a separate response in their survey. Previously, they were either buried in hematology or immunology. So these numbers aren't too bad, but when we then look at retirement rate, anticipated flow cytometry retirement rate over the next 5 years, the situation verges on dire. Overall, for all staff, we're looking at 18%. If you're looking at bench staff, 17% anticipate retirement. So this is people that are towards the end of their career. And for supervisors, it's over 1/5 of the total supervisor population in flow cytometry labs is expected to retire in the next 5 years, which tells us that we are definitely I'm not concerned at essentially having too many staff that the labor pool is too dilute. I'm actually more concerned about there being too few. And certainly the fact that many of you answered resource when asked about the most pressing, what was the most important of the 3 R's, points to the fact that you also understand essentially the looming crisis in lab staffing. So we're going to ask this question. In your laboratory, which of these options is the closest labor consumables expenditure mix? Do you spend more on your staff, on labor, or do you spend more on consumables? So your mix is majority consumables, 50-50, and the majority labor. And I will say that it's not uncommon that labs don't know this. It's pretty common that people don't actually see their expenditures. All right. However, most of you either do have access to that information or understand that, in fact, you spend a lot more money on labor than you do on consumables. For those of you who don't think you do, it would be very surprising if you are able, either you're in a geography where labor is very, very inexpensive, or is in some way being subsidized. So a lot of people have actually looked at this question. I particularly liked this paper because it actually broke it down into these nice little pie charts and actually showed the change from 1991 to 1990. So in 1991, and this is all laboratories, not just the very labor-intense laboratories that do a lot of laboratory-developed testing. So a laboratory-developed test almost always has a higher labor component. Reagents may be more expensive, but relatively speaking, the labor component will also be very high. So in 1991, these were kind of equal, labor and reagents. We have overhead here, but people were spending a little bit more on labor, but not an enormously larger amount. But if you then move up to 1999, over on the right, you'll actually see that labor has increased to 65% of the overall OPEX, and equipment and reagents have dropped to 15%. And you'll notice also that the overhead has stayed essentially flat. So if we then back out that overhead number, this change in the labor contribution to your laboratory OPEX becomes even more striking. And I'll point out that you even have equipment and reagents, not just reagents, that are in that 19% that's hiding behind that little piece of pie there. So it gives you a sense of just how much money you really are spending on labor. So that actually completes our review of the 3 R's. We talked about reimbursement, regulatory, and resource. And I'll go ahead and point out that at the end of the session, we are going to have a Q&A. And so go ahead and start right. If you had a question that you want to ask, don't worry. We don't know. I don't know who's actually asking the questions. Feel free to post one if you have any questions about what we already discussed, because we will want to be having that session at the end. So the next point is actually we're going to be discussing this concept of the 8 wastes. So this may be a new concept to some of you, but to many of you it won't be, because this idea is increasingly being used in context outside of manufacturing. Now, it actually does come from a manufacturing context. The 8 wastes is actually part of Toyota lean management, and at first you think, well, they're wastes. Why do I need to figure out which one they are? Well, it basically just helps you to take a systematic approach to what your wastes are. Is it a question of maintaining too much inventory and the associated costs? Are you moving stuff around too much? Are you making defective merchandise or defective products that you then have to scrap? So if we take the same concept of 8 waste and apply it to a laboratory, suddenly it resonates. So in this case, for instance, if I look at defects in the setting of a flow cytometry lab, here are some of the errors that can be made, and I'll point out that many of these have to do with the fact that there are humans involved. So for instance, you can add a wrong reagent, you can prepare a cocktail incorrectly, you may have switched the samples. This is the absolute worst error you can make in any laboratory setting, as you know, and of course transcription errors are very easy to make. In the case of non-utilized talent, this is considered a very specific type of waste, and it's one that should never be ignored. So this includes high-skilled techs performing low-skilled tasks, and the fact that it was that opportunity cost. New and improved assays go undeveloped because you were too busy pipetting or dealing with or doing your inventory management. Here, this particular one, motion it tends to resonate with many techs. If you're having to transcribe, that's unnecessary motion. If you're pipetting, that's unnecessary motion. If you're opening and closing containers and refrigerators all day long, that is unnecessary motion. So when we're talking about 8 waste, it's just a very helpful way to think about your operation and really drill down on them, because sometimes categorizing them really highlights them for you. You realize, gosh, we spend a lot of time doing this. I wonder if there's a way to get rid of that. Now, the next thing we're going to do is I'm going to show you some tools that you can use for assessing waste. So if you're going to assess your waste in the laboratory, and there may be some of you on the call right now that are thinking, oh, we have no waste in the laboratory, and we never make mistakes as well. I hope that none of you think that. All laboratories make mistakes. The question is how good you are at catching them. And essentially all operations have waste. Even any company or any laboratory that thinks they've drilled all the waste out of their operation needs to go back and look at it, because you can always take more waste out and improve your efficiency that way, and typically improve the quality of your product as well. So in order to identify waste, typically you have to observe. You really have to see what people are doing. Simply talking to people about it or reading a procedure manual will not usually show you where the waste is. You want to document it so that you will be able to prove to others that you found it, and also be able to have something to measure against. It's your jump-off point. It used to take us an hour and a half to do this task. If you want to be able to say, well, we made these changes, does it take us more time? Does it now take us less time? Does it take us the same amount of time? And ideally, you do want to be able to identify things that you can quantify. How many minutes does it take? How many steps does it take? How many heading steps? How much inventory do you have on hand, and so forth. Then once you've done it, you want to prioritize. You want to identify things you think will have the biggest impact and address them first. And this is typically called an 80-20 rule, and I'll show you in a minute how you figure that out. Then you want to implement your countermeasures and then sustain and track relative to whatever you'd identified earlier as something measurable that you'd be able to track against. So this first step is identifying waste. A really useful way of doing this, which might seem kind of useless but tends to be extremely illustrative, is something called a spaghetti model or a spaghetti diagram. And what we have here is just a floor plan of a workspace. This is manufacturing. I have 2 employees here, and we're literally just drawing them. You actually sit there with a piece of paper, and you actually draw their movement, and you can see where they go. And if they do it again, if this guy were to take this trip again, you would draw it again. And you can also measure the amount of time or the number of footsteps, or approximately. And you can also often do that by counting the tiles on the floor as a really quick and dirty way of measuring distance or footsteps. So this is a spaghetti model, and this is a graphic, a cartoon of a spaghetti model. I'm now going to show you a real spaghetti model, and this is actually in a laboratory. And if you look, for instance, this would be a pretty typical laboratory space. And each of these colors is a different individual. So as you look at the bottom diagram, the spaghetti diagram that's indicated in blue, that individual spends most of his time in one area. And you could actually drill down on just that one area. And you can see there are all the different strands of spaghetti having to do with the repeated trips that somebody takes. And this can be very helpful in figuring out that, oh, you actually could, by moving something from one room to another, you could actually make it more efficient. Now, you can also follow, instead of following people, you can actually follow a product. In this case, it would be a sample typically, or if you were, say, manufacturing some reagents or something, you could follow them. So another way of doing a spaghetti model is to actually follow either people or products or samples around and see what happens to them. Once you've done that and you've actually identified what looks to be a hot spot, you can actually prioritize. And this is called a Pareto Chart. And Pareto Chart is a really useful way of essentially ranking the problem, the issues you've identified, the sources of waste or complaints in this case. This is actually done for a restaurant. And this can be very helpful in terms of separating out what is actually important. If you would ask the owner of this restaurant why customers are unhappy, he might have said, because he might have gotten a few complaints to his face about it, that, oh, it's terrible atmosphere. I need to improve my atmosphere. But if you actually go and get data and ask your customers what they didn't like about your restaurant, it turns out that many of them felt it was overpriced, and many of them felt they were small portions. And if you then rank these in decreasing order, and then actually what we've charted here is a cumulative percentage. And basically when you pass the 80% mark, this is that 80-20 rule, you stop. This stuff you're not going to worry about right now because it's just not worth spending time on, but these are expected to have a high impact on, in this case, the restaurant, but if we had developed in a lab, on your overall reduction of waste in your operation. All right, so our next option, or next topic, will be to look at some streamlined workflows. All right, so, and this is where we start to implement countermeasures. So in this case, I actually have diagrammed the workflow. And again, it's very helpful. This is informed by an earlier spaghetti diagram. And so for those of you who may not be involved in flow cytometry, it's a laboratory-developed test. And in order to do the testing, you actually have to buy single-color reagents and combine them in order to generate results. So the steps involved in this are, let's say you want to do some testing somewhere, maybe not you, but somebody in the lab had to check the inventory of the single-color antibodies, discard outdated single-color antibodies because they're only good for a limited period of time, and then document, of course, that you've discarded those. If it turns out you need additional single-color antibodies, they have to be ordered. And then once they're ordered, they have to be received, they have to be inventoried, and you have to know where they are and their expiration date when you've opened them and so forth. The usual good laboratory practice. When it's actually time to do your testing, we move over to the right side of this chart, of this diagram, and you essentially have to assemble your single-color antibodies. And historically, many labs did this, and many labs still do. You would actually pipette single-color antibodies. So this particular lab is doing 4 antibodies per tube because they're doing 4-color flow cytometry, and to do their particular panel, they require 14 tubes. And this actually reflects exactly what I was doing in my laboratory when I was doing 4-color flow cytometry. My typical panel was 14 tubes. There was a lot of redundancy, and it was 4 antibodies per tube. So if you look at this, I'm going to, anywhere along here, I could identify sources of waste around any of the steps in this process. But in this case, I'm highlighting this last one, this pipetting of single-color antibodies, because this is actually a lot of work because it's error-prone. It's being done manually. It's repetitive motion, so the person who is doing this is having to actually pipette each of these antibodies individually. So that's actually 56 different pipetting steps, not counting taking off reagent bottle tops and putting them back on, and of course is time consuming. So let's look at a potential countermeasure for doing that. And the countermeasure I'm adding is this one here. Instead of having the tech actually do single shots, instead we're going to prepare multicolor cocktails of single color antibodies. And so in this case, I'm going to prepare 14 different cocktails, and so it's one cocktail per 14 tubes. So we've reduced the number of pipetting steps from 56 to 14. So still quite a bit of pipetting, but certainly better than it was before. So that's certainly an improvement. We're going to color it green. Visual management is also a nice thing to have in the lab. But it turns, and it's great because there is less opportunity for error, so you're not going to make single color mishits, less repetitive motion, and it's going to be faster. However, by doing that, by adding this step to our workflow, we've actually created some additional work as well. So you're going to prepare these multicolored cocktails with single color antibodies in your own laboratory using wet reagents, but that means you're going to have to QC them. And in some instances, depending on how many cases you've got, you may be having to discard outdated cocktails. In addition, the preparing of multicolored cocktails with single color antibodies and so forth, that actually doesn't fix any of this earlier workflow. Everything to do with the receipt of your single color antibodies and this part of your workflow, which is, trust me, quite cumbersome, is still not addressed. Now, and you say, well, okay, that seems like a wonderful thing to address, but I don't currently have the resources to do this. Is there another way to do that? So you could actually look and try and actually directly fix these problems, or you might be able to fix another problem. And that is, we can actually, instead of having 14 tubes, we can go to 4 tubes. And the way you do this is to switch from a 4-color instrument to a 10-color instrument. So there's more than one way to drill down on the issue. One would be to do away with your single color wet reagents. The other is to move from a 4 or 5 or 6 color platform to a 10 color platform. So these are all examples of countermeasures that you can put in place to fix your workflow. Let's see. Oop. I'm actually going to move to this slide. All right. So we're finishing up now. I'm just going to point out that workflow is extremely important in your lab. It's not very difficult it's actually not that difficult to assess. And you can actually simultaneously address all 3 R's, regulatory, reimbursement, and resource by looking at your workflow. And typical low-hanging fruit that almost all laboratories that are doing especially laboratory-developed testing can consider is look for opportunities to engineer out manual steps and transcription. These are almost always associated with are very labor-intense, and they are enormous opportunities for error. Decrease batch processing where possible. Batch processing is something we avoid in industry, I can tell you that. And if you can avoid it in your lab, that is generally better. And anything you can do to simplify inventory management. Inventory management is the hidden time, sort of time suck in your laboratory. So anything you can do to limit that is going to pay off. In the setting of flow cytometry in particular, there is something you can do that we actually address, was to maximize your multi-parameter capacity. So if you can actually do fewer tubes, you decrease your labor. At the same time, although I wasn't emphasizing decreasing your consumables cost, by having fewer tubes, you actually have require less antibody redundancy, and overall will use less reagent. And then there's actually a concept of stacking markers so that you can actually get even more markers in a tube and thereby maximize your multi-parameter capacity. All right, so the 3 R's are closely related: reimbursement, regulatory, and resource, and you actually can address all 3 of them simultaneously by addressing your workflow. So I'm going to stop there. We have a few minutes for questions and answers. So we're going to open it up. Christy, you want to handle this?
Absolutely, and thank you for that informative presentation, Dr. Holden. As she mentioned, it's time for Q&A, and if you have a question you'd like to ask Dr. Holden, please do so now. As a reminder, just click on the green Q&A button at the lower left of the presentation window, type your question in the box, and click the send button. We will answer as many of your questions as we have time for. Okay, let's get started. Our first question, Dr. Holden, do we know when the FDA's final LDT guidance will be released?
Oh, yeah. So, so the draft guidance came out in October of 2014. We don't know for sure when it's going to be issued, but we have heard that it is likely, a number of sources say they expect to have at least a next version, if not necessarily a final version, out by the end of 2016. I will say when I talk to people at the FDA, they can't say one way or the other, but they seem to nod their heads suggestively. So we're hoping that by the end of 2016, we know more about what that final guidance from the FDA will look like and what we're going to be up against.
How can Medicare justify cutting reimbursement for some tests like flow cytometry so sharply?
Well, so they justify cutting a lot of things based on the fact that they only have so much money and they need to spend it. And in the case of flow cytometry, they have a strong feeling that in some care settings it's being over-reimbursed. There actually is an issue with the CPT codes, and this especially in the case of reference testing. The CPT codes actually had a kind of built-in time bomb in that too much of the consumables expenditure was actually loaded into the licensing solution. Some incredibly large amount of the process expenses were actually somehow loaded into the licensing solution. So when they actually, the RUC, which is the agency that looks at practice inputs, when they looked at that, they said, well, there's no way that people are using 100 mils of licensing solution, or actually it's like 500 mils of licensing solution, and they cut that, and it cut reimbursement. We've actually been pushing back. A lot of stakeholders are pushing back on this, and we've actually managed to slow down the pace of cuts, but we do anticipate that we'll still see them.
How can we predict the risk categories for the LDTs that we currently perform?
It's going to actually, in order to do that, we're going to have to see what the FDA's final guidance looks like. Right now, we know that certain types of testing are higher risk and will have, will be assigned to certain risk categories, and those are infectious disease testing for things that are highly infectious and things that affect the blood supply. But other than that, it's actually, we're not really going to know until the new version comes out.
Now, Dr. Holden, if I adopt workflow improvements that eliminate labor, aren't I putting my own job at risk?
Yeah, I think that's an interesting question. A lot of people, I think, when you first think about, well, less labor, that's less of me if I work in that type of setting. And I think the key there is to realize that the amount of labor available, that pool is shrinking, and that we don't expect to have less work. We actually expect to have more work. And so I think, although it's something you might think about initially, it's probably, I don't think anybody is going to be put out of work as a result of improving their workflow. And actually, you'll be able to do essentially more interesting work. Very few of us, especially those, I know a lot of people here are in flow cytometry, very few of us want to do more of the low-value tasks. We want to do more of the intellectual tasks.
Dr. Holden, can you explain the difference between 3 R's and Lean Six Sigma?
Okay. Well, 3 R's is nothing to do with, it's not part of Toyota. And actually, I see the person asking the question, that is actually not from the U.S. So the 3 R's actually is historically a term that's used to describe education. So it's actually part of our lexicon. So we actually sometimes use it as shorthand. It's actually it's reading, writing, and arithmetic. Only 2 of which actually start with the letter R. So it was just a shorthand for the fact that these 3 things all start with the letter R. So Lean and Six Sigma are, Lean is actually part of the Toyota management style. Six Sigma has to do with how tightly controlled a process must be. So if a process is, if errors very rarely occur in a process, that is to say, usually something that's very highly automated, that means you actually have to do less control in order to ensure that you are doing it correctly. For something that's highly manual, you're going to actually have to have a lot of controls in place to make sure that you haven't made any errors. And so in a typical lab doing high complexity testing, you're going to have a lot of controls along the way. You have them even if you don't necessarily know, because you have multiple people looking at the results all along the way. So the 3 R's is nothing official. Lean and Six Sigma are widely recognized terms that are related. Lean is kind of a way of getting to Six Sigma. When we say something is we'll describe something in terms of the number of sigmas. So 3 sigmas is essentially not very well controlled. 6 sigma is very much in control, and you don't have to do a lot of checking at the end of the process to make sure you've done it correctly. Next.
What is the ratio of labor to consumables today? It was noticed that your graph showed 1999, which was more than 15 years ago, and now there is more automation available for testing, so demand for labor has decreased.
Yeah, and I wasn't actually able to find, so this particular paper I used because it actually had compared the 2 data points. There is literature out there that does describe that shows very similar numbers as recently as, say, 2008, 2009. So even though we are getting increasing automation, a lot of that automation is happening in the setting of, say, hematology laboratories and immunoassay. For labs that do laboratory developed testing, like flow cytometry, there is not enough automation to have a big impact on it, and it's one of these where I would actually love to actually publish data from a flow cytometry lab and actually see what those numbers are. Is that I haven't been able to find a reference that specifically drills down on that. So if anybody wants to send me that reference, that would be fantastic. Let's see. I'm looking at okay. Oh, here's okay. I'm asking some other questions, so I'm gonna. Okay. All right. So, Christy,
okay,
I just sent you a couple. Yeah.
Yes, absolutely. Let's get to these. We still have time for a few more.
Okay.
Dr. Holden, did Obamacare affect reimbursements to hospitals?
So Obamacare has not in and of itself made a really big difference. All of the trends that we're seeing were in place well before Obamacare. It has not, certainly we don't see it as in and of itself making a big difference. It's more on the question of how many patients are covered, which of course speaks to the intensive care and so forth. Most of the patients that are entering the system through Obamacare are actually younger patients who are generally speaking in better health. So they are not yet consuming that very high intensity health care. Obviously, if you're providing more care in general, there's an issue there, but it's not had a direct effect on the reimbursement, as far as I can tell. It actually has encouraged, there were some other things like meaningful use that are encouraging labs to, say, have electronic health records in place and so forth. But in terms of the actual reimbursement, all of that was going to happen anyway.
Have there been any discussions of artificial intelligence improving lab settings, decreasing labor costs, increasing productivity?
Absolutely, absolutely. So AI is actually kind of one of the holy grails in my particular area in flow cytometry, because we actually have to use a computer program to analyze the data and interpret it. This question of what we call assisted analysis is one that everybody who doesn't do flow cytometry and sees how we do it automatically says, oh, well, a computer could do that for you. And then you have to explain how it is that so far we're not quite there. But I think we, in the not too distant future, are going to see not necessarily automated, but certainly assisted analysis. And of course, we do have automated analysis for some of the lower complexity assays, like stem cell and lymphocyte subsets.
We have great questions coming in, but it looks like we have time for one more. Let's end with this one. What are the key factors, in your opinion, that make it hard for lab personnel to be recruited and retained? Is it salary-based, lack of professional development? What are your thoughts?
You know, I think it's both. The salaries actually are not bad. I mean, considering for the type of work you're doing, it's not bad. However, that labor pool, because they're typically so highly skilled, can be recruited by people that are willing to pay more. For instance, my company. So that is an issue. The other is, you're right, in terms of professional development, you know, if somebody who's, no matter how talented you are, your positions in a clinical laboratory are essentially, you can be a bench tech, you can be in a supervisory role or a technical specialist, and after that, you're really, you know, there's, you know, even a really large, say, university laboratory will typically only have one overall manager, and then you're looking at going into hospital administration. So the opportunities for professional development are not great, and part of that has to do with spending more time doing essentially manual labor rather than intellectual labor. We're seeing a decrease in the number of schools. It's multifactorial, but it definitely is. We're not getting as many people entering the lab personnel workforce, and they are also, by and large, are not as highly skilled coming in. We found that there's a bit of an educational gap as well. So that's where we are.
Dr. Holden, thank you again for this great presentation and excellent Q&A session. Do you have any final comments for our audience?
I'd just like to thank everybody for attending this morning. This has been a pleasure, and I hope you go back and look at your workflow.
Thank you once again, Dr. Jeannine Holden. I would also like to thank our sponsor, Beckman Coulter, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 1st, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. We hope you share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us. We hope to see you again soon. Goodbye.